In this video, we're going to be looking at a specific operon, which is what we call the Lac Operon. In bacteria, this is the operon that regulates genes that are related to lactose metabolism. Lactose, of course, being a disaccharide that is particularly common in milk-based products. So if we look at the different components of the Lac Operon, we see that it has all of the hallmarks of your standard operon structure, with P, of course, being the promoter sequence, and this is where the RNA polymerase will eventually bind and transcribe the 3 genes that are located within the actual genomic section of the operon. And these genes, rather unimaginatively, are what we call LAC-Z, LAC-Y, and LAC-A. And again, all of these genes are directly or indirectly related to the metabolism and energy production using lactose. Now, the characteristic regulatory sequence of operons, however, is of course the operator, and the operator is of course the location that repressor proteins or any transcription factor will bind. And in the case of the Lac Operon, the transcription factor that we're going to look at is indeed a repressor protein, and here we can see that this operon is in the off position because the repressor protein has bound to the operator, which means that it is impossible for RNA polymerase to either attach itself to the promoter or to progress past the operator towards the genes, and therefore transcription is not able to begin at all. So now that we have an understanding of the background structure of the operon, we can take a look at what each of the genes regulated by the Lac Operon do. And as we've already established, each of these genes is somehow connected with lactose metabolism, starting with LAC-Z, which codes for a digestive enzyme called beta-galactosidase, although we humans know this enzyme more as lactase, the hydrolytic enzyme that breaks down lactose, a disaccharide, into the 2 monosaccharides, glucose and galactose. However, as a disaccharide, we know that lactose is not able to cross a cell membrane without the aid of some sort of carrier protein, and that's exactly what LAC-Y is, which is beta-galactoside permease, referring to the carrier protein with an active site specifically to allow lactose to be able to enter the bacterium and be metabolized appropriately. The final enzyme is an enzyme called transacetylase, which has a complicated function relating to lactose's metabolism in cellular respiration, and therefore being able to use lactose as a source of ATP. But we can see that all of the functions, especially LAC-Z and LAC-Y, are directly related to lactose metabolism, which means now we can talk about the actual process of regulating the operon using the operator. So here we can see that the default position of the operator is in the off position, because as long as the Lac repressor is being produced, its active site has a conformation that allows it to bind to the DNA of the operator, thereby stopping the promoter from reaching the beginning of the genomic section. This means that we're going to need a substrate, which, as we talked about in the previous video, is what we call an inducer to bind to the allosteric site on the repressor protein, cause a conformational change that will result in the repressor protein falling off of the operator, allowing the RNA polymerase to bind to the promoter and begin the process of transcription. Now, the simplified form of understanding this operator simply states that lactose, the target of our digestive enzyme, and this transport protein here is the actual inducer, but that's not completely true, because we see that this chemical is not lactose proper, but allolactose, which is what we call a lactose metabolite, meaning that after lactose is brought into the bacterium, it will be modified to this form allolactose, which will make it easier to break down and easier to convert into ATP. And it also happens that allolactose, rather than lactose, will actually bind to the allosteric site of the Lac repressor, causing it to be detached, that being causing the repressor to detach itself from the operator, and therefore allowing RNA polymerase to begin the process of transcription of the Lac Operon genes. So if we want to summarize the regulation of the Lac repressor here, as more and more lactose enters the bacterium, meaning that as the concentration of lactose inside of the bacterium increases, more of that lactose is going to be metabolized into allolactose, causing its concentration to increase as well. And the more allolactose we have, the more likely it will be to bind to the allosteric site of this repressor protein, causing it to fall off and thereby increasing the level of transcription of the genes regulated by the Lac Operon. Now, of course, all of this also happens in reverse. So as the concentration of lactose decreases inside of the bacterium, that means that there's less lactose to be metabolized into allolactose, meaning that this concentration of our inducer molecule will decrease, meaning that it is much less likely that allolactose will attach to the allosteric site of the repressor protein, and therefore our repressor protein becomes active, and therefore this results in an overall decrease in the activity of transcription of the Lac Operon's genes. In the next video, we're going to look at the opposite situation in terms of looking at an operon that is not off by default, but is rather on by default, and where the inducer is actually a co-repressor in that it increases the ability of the repressor protein to suppress the process of transcription.